All right. Okay. Exam to review. All right. So brief introduction to the two topics we've been working on for the last 5 weeks. Am I recording? Yes. Yes. Am I? No, it's not capturing my audio.
Is it because you didn't break it on the microphone?
No, I hope not.
What? Why are we laughing at that?
Is it capturing? Okay, it's capturing now. Okay. All right. No. Is it capturing or is it not capturing?
I can tell.
You gotta look at the little microphone.
It's capturing.
It is? Okay.
See if we-
All right. Bro, why is it doing this? We're gonna pretend that that thing is not on the bottom left.
you can make it more sensitive.
It's like to the max.
Talking to the computer.
No, it's like as loud as it can.
Oh.
Yeah. Okay. So innate versus adaptive systems. We've been talking about this for quite a bit now, even though we moved on to the microbiome topic. I'm gonna hide this tray because it's bothering me. Anyways, so for our innate immune system, we have the most of the myelogenous blood cells. So starting with the myelogenous progenitor, and then going on with your megakaryocytes that then become platelets. You have your red blood cells, your erythrocytes, you have your mast cells, and then you have the cells that differentiate from the myeloblast, which are your three granulocytes, your basophils, your neutrophils, and your eosinophils. And then you also have your monocytes, which then differentiate into macrophages. And for our adaptive immune system, we'll have both our lymphocyte types and also our plasma cells, which differentiate from your B lymphocytes. So innate versus adaptive immunity. This is a chart that we were looking at last time around also. And so if you can look over here upon the primary infection, you'll have a little lag phase on your adaptive immunity, while your innate immunity will be almost instantaneous. And as you'll see, you'll have a lesser magnitude of response when compared to your adaptive response, but your adaptive response takes longer in the first stage. And so you can notice the long lag phase. And then on the second one right there, on the second infection, you can notice that there's a shortened adaptive response because it's the second time around that you're seeing this pathogen being invasive. And so your cells already have memory of what those pathogens are. So a little breakdown of innate immunity. So mostly all multicellular organisms have some sort of innate immunity. The different kinds of cells, adaptive or adapted through evolutionary history for this quick response. And it's like fairly quick, like less than a day to start reacting to pathogens. What ends up happening a lot of the times is like our bodies have been exposed to so many different pathogens that, your innate response isn't really like compared to your adaptive response is not as effective. But it's mostly because, with different like strains of the flu, for example, like sure, it's a different virus, but then it's just an evolutionary adaptation of like an older flu strain. So your body can kind of also like recognize those pathogens. And so your innate immunity is your body's first line of defense. The main pathogen blocking response that we have is the skin. It's our largest organ and it prevents entry from the body unless you have abrasions or cuts, which is then when the rest of your innate immune system starts to act. And right there, the response happens quickly, and the mechanisms are the same for all invasions, so there's no adaptability to like the pathogen is gonna react the same way. The inflammatory responses are gonna be the same. And so you can have physical barriers like the skin and then the different immune cells, and there's a little breakdown over there. That's not all of them, there's most of them over there, but you don't have to get into that much detail. And then we have adaptive immunity. So as the name suggests, it can adapt to prior invasions. And these are mostly your B and T lymphocytes, although you also have your assistant cells like your dendritic cells, your macrophages, and your plasma cells. Also, the B cells also serve as antigen presenting cells for your T cells. But that's a story for different for later. And so you have recognition and targeting of identified exogenous particles. I said only exogenous particles here because we haven't gotten into like autoantigen and autoimmunity. And so there's going to be hypervariability in the antigen binding domain of antibodies. And this leads to a really, really high number of identifiable particles. According to PubMed, it's 10 to the 18th power, which is 1 quintillion. So that's like a lot of possible recombinations through a process that we'll cover in just a second that you guys love. And here they're reliant on memory cells, which make the process easier and faster, but also like you're reliant on your body having storage of the previously encountered pathogens. You wanna get this one?
Yeah, because okay, close silence. Thanks. So antigens are basically any sort of small particles that can be identified by the adaptive immune system. You want these to be things that are not self, right? Because you don't want to attack yourself, but we do have things called self antigens, which is a whole different story that we'll get in. Are we going to get into that?
What?
Self antigens and stuff, or is that autoimmune?
We didn't cover it, but I didn't make a slide.
That's fine then. Yeah. So these can be sourced endogenously and or exogenously, right? Because it's either yourself or it's outside, or it's just for our purposes, it's anything that's foreign and we need to get rid of. They can have a variety of different identifiable sites called epitopes, and that's exactly where like the antibody will bind. So that one, you can see there's different epitopes with different shapes, so different binding properties, right? And you need different antibodies with different binding properties to bind those different epitopes. I mean, I guess you don't necessarily need to bind all of them on an antigen to elicit a response, but the more binding you have, the more response you're going to get, right? Well, we might talk about this in a sec, but when you have T cells and B cells that are activated, they don't just see an antigen bound to a single antibody and then take it in. It has to be like this cluster of antibodies that surround an antigen because the Fc receptors need it to engulf and then do whatever it needs to do inside the cell. So more antibody binding you get with different epitopes, the better. And that's where the recombination comes in. Antigens can be literally anything. It could be, doesn't have to be living. It doesn't have to be like multiplying. It's just something that's not self in the body. That's my section. You want to do this? You're better at this. You're better at this. Huh? All right. Antigen binding structures. So we have BCRs and TCRs. So BCRs are antibodies which are membrane bound. So you have let's see here, because I didn't make those. So your B cell receptor produces these, like, I like to think of like the ones. This is like an antibody when I think of it. Like this is what I think of. You have your heavy chain, your light chain, and your variable regions, and your constant region. And that's typically the structure of a B cell receptor. A T cell receptor is kind of different, where you don't have that like Y shape. Instead, it's kind of just you have your variable regions just straight on your constant regions, and then it's just basically made up of an alpha chain and a beta chain. And it's important to know that TCRs aren't antibodies, right? It's the BCRs that are more that are considered antibody-like. Anything else I'm missing?
The T cell receptors don't diffuse away from the cell.
Yeah, they stay there. That's important. Cool. B lymphocytes. What do you want to say here?
Well, that's just a visual. There's the next slide.
visual. Okay, so B lymphocytes are involved in antigen presentation. So this is when the B lymphocytes will take in the antigen, dismantle in a way that kind of preserves some certain parts of the antigen so that it can be presented back outside the cell. And this allows it to release antibodies, and it's part of the inflammatory response. These are generally free flowing, so they're transient. You know, they go around everywhere and perform functions where they need to. They can differentiate into plasma cells, as you saw in that little graphic that John showed at the start. That's kind of like the end of that whole chart. And it's not reliant on memory cells, right? These are just, you know, senses the antigen, breaks it up, release every antibodies, right? Yeah,
And they're still very specific to a specific antigen, right?
the B cells, I don't think they are.
Well, each receptor,
Yeah, each receptor,
but not the cell.
not the cell. Yeah,
But they're all gonna be producing different antibodies simultaneously.
Okay, so it's not just one B lymphocyte for one antibody.
No, it's like one B lymphocyte for as many antibodies as it can carry. But like the antigen binding sites usually like 6 amino acids like long.
yeah.
So it's like tiny.
Shouldn't hold a ton of different ones on one, so.
Okay, so one B lymphocyte takes a bunch of antibodies, and then the antibodies are favorite that's like selected for,
What do you mean selective?
Then the antibodies what?
like against, like, the memory Bs.
So, I mean, it happens by chance and you'll randomly have like an antigen like diffuse into the receptor, right? And then once that does happen, it takes the receptor in and then it like presents it to other cells like the plasma cells, the mast cells. And then the mast cells can then present it to like the T lymphocytes too. But then it causes an overproduction of whatever antigen if it's like recognized. And then they release it. Okay, so this is just a little slide for clarification of receptors versus antibodies. So the receptors are membrane bound, which means they don't diffuse away. And they promote the replication of antibodies, like we were just talking about, the antibodies that do bind antigens. They trigger inflammatory responses, and then they can activate other cells, like your mast cells, your macrophages, which then leads to activation of T lymphocytes. While your antibodies are free flowing and they're diffused away from the cell, and they're known as immunoglobulins, and we'll cover the types in just 1 second. We don't have to go into like that much detail other than like the reason why there's different ones. But yeah, that's the major difference. So antibody structure, as Eren was talking about, you're gonna have 2 identical heavy chains, 2 identical light chains. The outermost part of the chains is where you're gonna have your antigen binding site. And all these chains are their own individual proteins, which together forms quaternary structure. And all these proteins are held together by disulfide bridges, and that's because it's the only R group covalent bond formation. And it holds together the proteins because it's a very strong covalent bond. And yeah, so you'll have your constant regions, you'll have your variable regions, and I don't know what I was thinking about on that last.
These are what.
We're gonna ignore that last sentence and I'll delete it before.
That makes sense.
Huh?
Well, it should be the other way. These are what, like, the end.
The
I see what you're
antigen.
trying to
I
say.
don't. I was probably like cooking and then forgot about it. Anyways, so then this is a visual of the chains of the receptors. So you have your heavy chains, and that's exemplified right there with the drawing, and then you also have your light chain. And the production of these is identical for both. So whatever you have on one side is exactly what you're gonna have on the other side too. And that's a visual of like all the constant genes, all the variable genes. On the image, it looks like it's one gene. It's not, it's more than one gene. It can be more than one gene, and we're gonna cover the RSSs and the 1223 rule. It looks like it's just one gene, it's not. It's multiple. It's just the type of gene. And right here, our favorite part about this entire topic, you can do it if you want.
Yeah, I can start it.
Yeah.
So VDJ,
I can
I've,
sit down if you want.
no, it's fine. I've said this like a billion times, but we'll go over it again. So VDJ recombinase is generally a group of proteins that involves activating genes or recombination activating genes 1 and 2 are called RAG. And these bind recombination signal sequences. So RAG1 will bind to the RSS surround the VDJ coding regions and induce a double strand break between the gene segments and the RSS. So this forms two things. One is a hairpin loop on the coding segment and a blunt end on the signal segment. So that signal segment, I don't know if you remember the video, it's like that, the first part they talked about, it kind of just like forms a circle and goes away. For our purposes, it doesn't matter anymore. What does matter is a coding segment and that hairpin loop. And you need this protein called Artemis to open that hairpin loop. Artemis is defunct in skid mice. So that's why they are skid. But we have it, it works, hopefully, and it will open that hairpin loop. When you get that opening of the hairpin loop, you get this protein called deoxynucleotidyltransferase or TDT. It adds additional nucleotides. And this is super important to the randomness of the variable region because it basically will go in, like add a couple like conserved like nucleotides and then just start like randomly like placing things, right? So the majority of nucleotides that it's going to place, it's like totally random. It goes by, I don't know what plays into it. But for,
Yeah, it's like it doesn't have a single,
yeah, it just,
it freestyles.
it's free. It's not bound to the laws of man. And it just, it's great because then you produce antibodies not only with like different sequence of nucleotides, but different number of nucleotides, right? It's not gonna vary by like a ton, but you know, that extra like 10, 15, whatever it is, it's gonna add to the variability and hopefully catch more antigens. So I think at the end of that, ends are just ligated, and you get this full VDJ region plus the coding, the constant region that's gonna be transcribed into the antibody. Yeah.
I think that it has to add them in triplicates. Well, it can add them in any random, but only the recombinations, the TDT when it adds it in triplicates, so it'll be in frame shift. Those are the only functional.
That makes sense. Yeah. Yeah. It has to have some rules, I guess. Yeah, and like John said, 10 to the 18th possible unique receptors, a lot of variability. Great thing for us. Cool. Oh, you're good at this. Cool.
Okay, so we were talking about the RSSs, so recombination signal sequences. You don't need to know all the jargon about it. Sure, they're conserved heptamer and nonamer sequences. What you need to know is that they're flanking each of the genes. And they're either gonna have 12 or 23 terminal base pairs. If you have two genes that have identical numbers of terminal sequences, so if it's a 12 and a 12, or if it's a 23 and a 23, it does not work. Hence, we have this thing called the 12-23 rule. As you can see right there on the visual, in the middle on the kappa chain, I've never seen kappa be used. Anyways, on the kappa chain, you'll have a V region that has 12 terminal base pairs, or on the H chain, sorry. You'll have a V region that's flanked by 23 base pairs, and you'll have a J region that's flanked by 23 base pairs. What happens then is that you need a joining segment that's, in this case, part of a D gene. And it's flanked on both sides by 12 base pairs. And so if it's 23 and 23, or if it's 12 and 12, you cannot have completed recombination. You need it to be 12 and 23 or 23 and 12. It needs to add up to 25.
You said we don't need to know like Captain America.
No. You just need to know RSSs exist and you can have identical.
What happens if when 12 and 23 are not working to each other the same? Or not?
It doesn't work.
Like, is the.
You added
gene marked for, like,
G.
different- Oh,
Well, the recommendation won't work. So you like evolutionarily something, yeah, you need a new gene. And it's, I mean, it's done through splicing.
Like, are they splicing? Or
I think it's
is it still
like-
DNA?
it's the recombinase complexes. And then, just to differentiate this, Laura was talking about somatic recombination, and that's the process of BDJ recombination. This is meiotic recombination. And it's the two big, kinds of recombination that we have in our cells. This only happens in our germline cells. That's the major difference. This only happens once, because we have 23 pairs of, chromosomes. Hello.
Hi.
We have 23 pairs pairs of chromosomes per cell besides our germline. In our germline, whoa. In our germline, we can only have one pair of each of the 23 chromosomes, for you to have, a pair or 23 pairs when, fertilization occurs. And so for every single disjunction event that happens, right, for you to have one chromosome per, or one pair per, cell, you need a, wait. Yeah, you need, a recombination event to happen. And so this is typically crossing over events. And so for the disjunction to happen in meiosis, you need at least one of these crossing over events. Or else it just does not happen. This is because it increases genetic diversity in our own genomes. And that's good evolutionarily because you're susceptible to survival in different environments by having genetic diversity. So like alleles or different types of, like sequences, right? Because you won't necessarily have the same allele be expressed in every single cell, right? What differentiates you from like every single other possible germline cell from like both your parents was the way that your genetic sequence was realigned, by this. And so that's why you are you. Anyways, very philosophical. Yeah, and so these are the different immunoglobulin types. I don't want you to know all this jargon about each of the types. You just need to know that they bind different epitopes. And that's why they have different names and functionalities. They are binding different epitopes. They are more specialized for binding specific epitopes. You don't need to know that IgM is produced first upon antigen evade. Like, you just need to know it's different. And they're binding different epitopes, and they have different functionality because of that. But you don't need to know the jargon. There's just 5 types. Capiche? Okay. Alrighty, you can get started on this if you want.
Is that just the visual?
Yeah, there's the other slide.
Right, lymphocytes. Like we said before, the T cell receptors are not antibodies, right? On your T cell lymphocytes, you'll have your MHC1. This is generally considered a CD4 positive T cell or CD4 positive T cell, sorry, have the MHC1. These are your helper T cells, and your CD8 positive T cells have the MHC2. These are your cytotoxic T cells. TCRs can't diffuse away from T cells, and the T cells themselves rely on antigen presenting cells to, and I guess in that sense, the MHC complex to, perform their function of whether it's the helper T cell function, the cytotoxic function. The alpha chain locus resembles the light chain of the antibodies. We saw the alpha chain and the beta chain. Remember that on the T cell, and the beta chain on the receptor. And the beta chain locus resembles the heavy chain. Yeah, I don't know what else to add here.
That's fine. Yeah, skip. So yeah, more specifically, you have like a bunch of chains. We're mostly interested in the alpha beta on the epsilon delta and on the gamma epsilon chains because they form pairs. You don't need to know about the zeta chain, like it's irrelevant. But yeah, so these cells are reliant on presenter cells. They can't just like diffuse around and like, oh, found this dude. So what ends up happening is these cells are being presented with the MHC complex. And so they need to be able to bind the peptide and the MHC at the same time, or else they won't work, right, by default. Because if you're being presented a complex and you can only bind partially, then you're not specific enough to perform your job. And again, CD4 positive and CD8 positive are the different types of T cells, and they bind specifically their own type of MHC. And if we set 4 equals to X, MHC2 is gonna bind CD8 plus, MHC1 is gonna bind CD4 plus. Okay. T cell maturation, so, we wanted to clarify, the process is like slightly different between T cell maturation and B cell maturation. Mostly it's because this happens in the thymus and, B cell happens in the, bone marrow. But the process is pretty similar and it makes sense why it's similar. Yeah, kombucha is disgusting. Yeah. And so for T cell maturation, what happens is the naive T cells are gonna be shipped off from the bone marrow where they're being produced to the thymus where they're gonna be matured into like functional T cells. And so this is a process of testing the random creation of TCRs. It happens very similarly to BCRs in the bone marrow. So there's the 3 different types of selection that we've talked about, but we'll cover again. So you have non-selection, so you have the T cells that just simply do not bind the MHC peptide complex. And these cells get driven into apoptosis almost immediately. And they are not selected for, therefore non-selection. Pretty straightforward. And then you have positive selection. So these are the T cells that can bind peptide MHC complexes. And so these are led to survival, so they constantly receive your survival signals, and then they are able to leave the thymus afterwards once they're mature. And this leads to replication and positive application of your TCRs. This is important because the odds of you running into a antigen that you know from that 10 to the 18th power number is good. And this is important because most of your functional T cells are gonna be required to perform their job of not letting you die because of pathogen invasion. And so these are selected for, so positive selection. And then you have negative selection. Whoa. And so these are T cells that are gonna be binding the peptide MHC complexes with too much affinity. So then the problem with this is that you have too high odds of these TCRs binding self-antigens, and we could do a little self-antigen talk in a second. And so the replication of this TCR could cause you to bind your own molecules at too great of a scale for it to be healthy enough to be maintained. And so these cells are also being selected against and are being driven into apoptosis. And so negative selection because they're selected against. And so we were talking earlier about antigens and Eren was explaining, antigens can really be anything. It could be glycoproteins, it could be proteolipids, they could be like, a bunch of different invasive like molecules. The biggest thing is that if we were to limit ourselves to have receptors that strictly do not bind antigens that we produce, so your self-antigens, that would lead us to have too small of a genetic variability in our receptors to fight off pathogens. Because it's not because we have similarly conserved genomes to like invasive pathogens that they're not invasive to us, right? And this is also a problem with cancer that we can't identify cancer cells sometimes because they're similar to ours. And so we were talking about autoimmunity versus autoimmune diseases, right? And an autoimmune disease is a disease that's nice. An autoimmune disease is a disease that is derived from autoreactivity to, whatever antigen. It could be like diabetes, like your GAD or your ZNT8s or your whatever other proteins that we don't need to talk about. But you can also have a similar like conserved kinase domain to, I don't know, a bacteria, right? And if we can't identify that, then, you have autoreactive TCRs and BCRs. It's important that they exist because the odds of us having a shared genome sequence with an invading pathogen is too high. And so if we were to like completely get rid of all of those, then we would just be at the risk of not being able to respond to all the invading pathogens that can invade us. And so by default, having autoreactivity while not having autoimmune disease is something that happens often. And if you remember, like a couple weeks ago, we were reading those papers and it says, oh, like a lot of people have autoreactivity, they just don't develop any sort of disease, and that's why. Okay, and then indirect autoimmunity. So that's that really, really dense paper that we covered. I think that the two major ones that you'll have access to paper during the exam. But if we were to keep in mind that, this thing also exists and, it's autoimmune disorders that are caused indirectly by autoreactivity. I think the example of myasthenia gravis, whatever, of the acetylcholine receptors being blocked and then phagocyted, engulfed by our cells. The best example of this, because you are not able to signal acetylcholine. And as most of us know, as you're struggling with 372, acetylcholine is a big neurotransmitter, especially for neuromuscular neurons. And so that's one of the main ones, the paper. I think the second easiest one that we could talk about is the activation of neutrophils. Neutrophils are part of your white blood cells, or aren't part. They are a type of white blood cell, and they can be activated by binding of their receptors, like some membrane receptor protein, that can be activated and trigger an inflammatory response due to the activation of these cells. Though you shouldn't like focus on like learning the entire paper. That paper is like super dense. Like don't spend your time on doing it. I think the first introductory paragraph that talks about like all of those 7 is good enough to like, as like helicopter knowledge. You don't have to like know everything about every single one of these. If you want to talk about blocking of enzymatic function, that can also work. Because the antibody is binding a specific part of the enzyme and it's not working anymore. Or you could talk about, mimicry of receptor activation by hormones. So that's when your antibody is going to go and bind a receptor and cause an activation of a like signaling pathway or something. Simply because it binds where it shouldn't bind. And that happens by chance in the generation of your TCRs, or in this case, BCR. But I wouldn't spend too much time on it. Just know that, indirect autoimmunity exists. Yes?
The point is that they all involve antibodies or self-antibodies, but not all of them cause an inflammatory, like.
Yeah, they're not like destroying the cells.
It's just blocking or.
Yeah, I
making
mean,
something-
cell lysis, yes, they're destroying erythrocytes, but every other one is like a secondary response to the antibody binding.
And not like a direct, like
Exactly.
typical immune response.
Exactly. Yeah.
Uh-huh.
Have fun.
Microbiome.
Have fun.
John hates the microbiome.
I hate the microbiome. It's too philosophical.
It's luckily this will be easier than everything else we just covered. All right, so microbiome. The essential question here is what essentially defines you as you, right? I've had some heated debates with John about this, but basically the microbiome can influence many parts of your body, including the way we think. So there's 30 trillion human cells, roughly 30 trillion, and there's roughly 38 trillion microbes, so we are definitely outnumbered. And it only looks like 8 trillion, but 8 trillion is a lot of cells to be outnumbered by. So that raises the question of, in our general makeup, like how much is actually us? What do you consider like human versus microbe? Are we, when we talk about ourselves, are we considering just our human cells with our DNA? Are we considering like everything that influences us? Because those microbes can, have disease-related effects, mood-related effects, and nutrient absorption-related effects. And like that's just a couple of examples that some of the papers talked about, right? That's my favorite photo I just discovered, where they're having a phone call, which like seems funny, but that's in a way exactly what happens. And I'm going to talk about it in a sec, unless John has anything to add.
I hate microbiome.
Yeah, so I thought, oh, right, before we go to that, shout out to Van. Van had this graphic on his slides review, which is cool, and I wanted to take it, and so I did. Yeah, what? Yeah, I didn't see this in the paper. I totally glossed over this because I saw it on Vans. I was like, whoa, that's sick. Anyways, John, what'd you do?
Why is it initiating the creative cloud? Dude, I didn't use Photoshop. Okay,
Go back, go back.
sorry.
All right, so you don't need to like memorize all these, but these are what the paper talked about, and there's some of them are really interesting, like the fecal transplantation. So you transfer feces of communities with these like microbes that I guess you need. And you can reintroduce or restructure an existing microbiome that's kind of failing. And failing based on your definition of failing, right? If you need certain microbes and those are gonna help you with your either like disease or daily functioning, then that is considered. Kind of gross, but it is. Diet and prebiotics. Probably you've seen, like those prebiotic yogurts and drinks and whatever, like those, what you take in can definitely influence, your gut and what exists there, what stays there, what multiplies there. Symbiotic microbial consortia. So this is a transfer of a group of isolates, and they select or design to promote a specific microbiota functions. You have an advantage is, it's a known composition of the consortia, and it can kind of introduce a self-sustaining community. But you have these replicating phenotypes that can emerge from the complex interactions. So I guess growing desired isolates in culture would be best before you go about that. I think that's what that's trying to say. You can have engineered symbiotic bacteria. So the transfer of bacteria that colonize a targeted site and are engineered like specifically to have a desired function. This is great because we can have specific results that, are proven and we know are going to work. But there's a limited ability to manipulate many of these microbiota species. So still in the works, definitely. And then microbiota derived proteins and metabolites. So this is like a direct supplementation with beneficial proteins. Pretty easy to prepare, and you can assess their safety pretty easily, just like simple drug production. But I guess determining and delivering the adequate concentrations to desired sites is difficult, because a microbiome kind of like we know where it is, but it's also everywhere, right? So it's hard to like pinpoint where you want it. Again, be familiar with these. You don't really have to be super experts on any of them.
Is that in the microphone? The paper?
Yeah, I believe so. Green point. This is blocking it, but I'll send you the slides after. It's a reference. So another important part of the microbiome papers is the gut microbiome brain axis, right? This is a bidirectional communication between the gut bacteria and the brain. That little phone call picture is kind of what this is referencing. So this is super important because it can alter the production of metabolites, neurotransmitters, and immune signaling molecules, which can potentially, very new research, but there are results coming out. Like you can have autoimmune diseases that are the result of this, like MS, ALS, any of the ones we talked about in the NDDs paper too, like those kind of like NDDs and autoimmune diseases. But also can affect your mood, which is super interesting, right? And it makes sense because if you have altered production of specifically neurotransmitters like serotonin, dopamine, et cetera, those kind of things, then it's going to change your mood, right? Maybe it might not be overnight, like just because you have this like new one single bacteria that's producing something, but like if you have colonies that are tailored to, outgrow others or kind of compete with others, and you have this predominant one that shows up that can influence, let's say, like dopamine levels in your brain, then that's gonna over time affect how you think and how your mood is. So there's definitely a lot of research going into first understanding how this exactly works and what sort of like specific microbiota are making like the specific effects in the brain, and then the therapies that can kind of combat that, whether that be like eradication or competition or something else. Yeah, that's it. That's all we had. So thank you for coming to our review. Good luck on your exams this week. I don't know if John has any other words before we send off.
Y'all will do great.
Great motivation.
Cool.